Hello everyone, welcome to today's Science of Childhood Cancer. I'm Charles Mulligan, Deputy Director of the St Jude Comprehensive Cancer Centre, and I'm very excited today to be able to introduce two rising stars of cancer research as our speakers, David Chen and Lileng Wang. Each speaker will present a 20-minute talk, which will be followed by a 5-minute Q&A session. And after both talks, Drs. Chen and Wang have agreed to stay for further discussion after the top of the hour. So we encourage you to submit questions at any time during their talks using the chat function and sending questions to all panelists. And if you would like to stay after the top of the hour for that extended discussion session. And so now I'm delighted to introduce our first speaker, David Chen. David is Associate Professor in the Department of Systems Biology and Director of the Division of Epigenetics and Transcriptional Engineering in the Beckman Research Institute at City of Hope. David received his PhD from the University of Rochester and subsequently undertook postdoctoral training with Scott Armstrong at Dana-Farber Cancer Institute and Memorial Sloan Kettering Cancer Center. David's work examines how genetic and epigenetic regulators control gene expression, the epigenetic landscape and genome integrity in normal development and cancer. And as you'll hear today, he's pioneered some innovative technological approaches, including gene editing to map protein functional domains and single-cell CRISPR screening. His work has been published in leading journals, many of which including Nature Medicine and Cancer Cell and Cancer Discovery, and he's received multiple awards, including an NCI Merit Award, Alex's Lemonade Stand Foundation Innovation Award, and an Ash Scholar Award. So David, welcome. Over to you. We're delighted to have you today.
Thank you so much, Charles. Yeah, and thank you so much for the invitation and the opportunity to present our ongoing work. So in today's talk, I think I will focus on try to introduce a new way of how to use the CRISPR genetic screen. So we start to develop a strategy to use CRISPR genetic screen to zoom into the protein domain function, and we call it high-resolution CRISPR tiling scans. So in today's talk, I will introduce two different pipelines. One is the more traditional survival screen. We use the high-density CRISPR tiling for a gene and therefore zoom into their domain behavior. And in the second part of the talk, I would like to introduce when we combine these high-density CRISPR tiling scan with the 10x genomics single-cell RNA-seq and create a new feature of the CRISPR genetic screen tool. So just one slide of introduction of the CRISPR. I think we don't need too much introduction, but just one thing we can see is the CRISPR system utilize the guide RNA with the sequence complementary to the targeted genomic regions, therefore recruit the Cas9 to exert the double-strand DNA break in the standard Cas9 condition. In this condition, the DNA break can cause the random repair and then sometimes cause the indel and then start to destroy the gene function if we put the guide RNA cutting within the coding region, for example, these exons, and could cause the frame shift of the whole protein. This is how we think mostly how the CRISPR can cause the gene deletion or loss of function. The CRISPR technology actually took a while to fly to here, even though it's become so popular today. At the very beginning, it actually took a long time for the early pioneers, like our Nobel Prize winners, that they spend a lot of time trying to characterize those repetitive sequence in the bacteria. Later on, find there's a signature genes called Cas is associated with this region. Later on, people find the Cas or Cas9 is actually an endonuclease that can be controlled by the RNA sequence, and they therefore target the particular DNA sequence for double-strand DNA break. And just within 3 years after that, people start to find if you put the Cas9 and guide RNA into our higher organism cells like eukaryotic cells, human mouse cells, we can exert the gene editing in our cells. And just within 1 year, the first genome-wide CRISPR genetic screen was conducted, and then there were actually few papers ongoing at the time. One of the pioneer at the stage, Fen Zhang, actually contribute in my mind a lot of this part of try to move forward the CRISPR technology to be a high throughput screen tools. What they describe is they use the microarray to synthesize the designed oligo that can target particular genomic regions, and then clone these oligos into the guide backbones vectors as a pooled library. Use the lentivirus to deliver into the targeted cells. Also with the Cas9 activity, you can harvest the early samples and unsampled and send for high throughput sequencing to deconvolute each guide RNA sequence and their frequency change. In this way, you can quickly test few thousand or even the genome-wide gene functions in multiple cell types. The power of this genome-wide research now is already go beyond extraordinary. Now we have more than 1,000 genome-wide CRISPR screens was already recorded and published in the consortium database. There are two major efforts along the way that try to move forward this genome-wide characterization of the gene function in cancer cells. One is the Achilles project, or now called the DepMap project, which they now already sequenced or screened more than 1,000 cell lines. If I just checked earlier today, it's more than 1,000 cell lines. They have used their genome-wide CRISPR library to screen, and therefore documents a lot of the gene behaviors and help us to give us the power of the first-hand evaluation of the gene role in different cancer types. Another consortium in UK, the Project SCORE, they also build a genome-wide library and screen in few hundred cell lines and some different tissues. So after all these efforts of genome-wide sequencing or CRISPR screens, when I started my lab in 2017 in City of Hope, I was concerning what the new direction I can move on with the CRISPR technology to move forward the field. So a lot of the data that I want to present today is actually inspired by this seminal paper published by Chris Veklich and then Junwei Shi in 2015 in Nature Biotech. What they describe is when we want to use CRISPR to knock out a gene function, typically we choose to put the guide RNA at closer to the N-terminal early exons. Therefore, when we cut the genes and cause the random repair, we have, if it causes the frame shift, for example, the indel cause the frame shift, it can cause the most of the protein to be loss of function. However, when they try to put the guide RNA in different portion of the gene body, they start to realize that it's not always the most N-terminal regions give you the strongest phenotype. For example, these are essential genes in leukemia. When they try to knock it out, not always the N-terminal guys got the strongest phenotype in terms of killing. They find that some guides are particularly strong in the cell killing, and they seems to be associated with regions that were described as domain. For example, this EZH2, the K27 trimethyltransferase, the enzyme core domain is actually located very close to the C-terminal. And then when they put the guide RNA over there, they actually exert the strongest killing function. BRD4, another example of the chromatin binding domains of the bromodomain that can recognize the histone acetylation. They find the guide, when the guide are hitting the bromodomain in this BRD4 protein, seems to hit a much stronger killing phenotype compared to just put at the N-terminal. So they come up with this rationale that if our phenotype require frame shift, in this case shown in the red color, to kill the whole protein function, our chance is about 2/3 of the chance can cause the frame shift of either allele in the random chance. If we need both allele to be frame shift, the chance is actually only 4 out of 9, or less than 50% of the cells can be double allele knockout. That is probably why when we try to create a knockout cells, one very important step is after deliver the CRISPR mutations, we need to single clone the cells and look for the double allele, both are knockout. Therefore, we can move forward with the project. However, they hypothesize that if the region of the protein is highly dependent on their conserved amino acid sequence to exert function, for example, in the enzyme core domain or in the chromatin binding module, they couldn't switch too much. Even the in-frame mutation shows in the blue color allele, they may still exert loss of function phenotype, and therefore the cells couldn't survive in a much higher proportion of the culture. So based on this idea, I try to move forward with our current CRISPR genetic screen pipeline and try to use the high density way to examine the domain function. So here I try to introduce the cell disease models that I'm studying here. It's the MLL rearranged leukemia. As Charles says that I was trained in Scott Armstrong's lab. This was one of the disease that we are very interested in and try to find the disease mechanism and any therapeutic opportunities in this pediatric leukemia disease. And this disease is typically you can find the patients are triggered by some of the one of these MLL gene translocation and cause the fusion oncoprotein with either AF9, AF4, ENL, and so on. And this disease is highly prevalent in the pediatric patients, and also it exerts a much worse prognosis compared to the non-rearranged patient. Molecularly, over the past 10 years, we have start to have a better understanding of how the MLL fusion proteins start to trigger epigenetic change in their target locus. For example, here, there's an enzyme called DA1L, which is so far the only enzyme we know can modify the histone at the lysine K79 position for methylation. And this H3K79 methylation is highly associated with gene activation or gene expression. Some of the target gene locus, including the MEIS1 or the HOXA9 HOX locus, are known to be controlling the leukemia function. Therefore, the hypothesis is the MLL fusion recruit excessive amount of DA1L activity to the target loci, put on too much of K79 methylation, therefore allow those pre-leukemic gene to continue to express. Their therapeutic approach was tested and tried by the biotech companies. For example, Epizyme, their pilot compounds finished the phase 1 clinical trial and showed the proof of principle efficacy. After a few cycles of treatment, the leukemia cutis was disappeared for this, for example. So for me, I will study this gene for a few years, and I'm curious what other regions in this protein, DA1L, is also important in addition to the enzyme core domain. So here we developed this pipeline. We use the microarray to synthesize all the guide RNA that we can target the coding exon of the DA1L. In this case, DA1L is about 1,500 amino acid, longer than 4.5 kb coding regions. We can design about 600 guide RNA to tile every possibility of if there's a pain, we put a guide RNA there. So our targeting density is around 7 to 8 base pair, or you can think about every 2.5 to 2.6 base amino acid, we have a guide RNA cutting on this DA1L. We clone them as a pooled library and then make them the lentivirus deliver into the F9 cells with Cas9. And then we harvest the early sample and end sample and sample sequencing as the typical way people do the CRISPR screen. And the result is shown in this way. So let me work through the result first. So in the x-axis, this is the peptide position of DA1L from N terminal to C terminal. And each green dot indicate a guide RNA and their cutting position. We already mapped them to the peptide position. The y-axis is the CRISPR score. In this case, it's the log10 scale of the endpoint versus start point of each guide RNA frequency change. So for example, it's -1 or -2 is equivalent to 10-fold or 100-fold depletion. The top of the rainbow score, the plot is the conservation score of the peptide, and the red is the more conserved region. The blue-green regions are more variable region across evolution. So here you can start to see that different guide RNA targeting different regions actually exert pretty diverse phenotype in terms of the cell survival. We start to see some cluster region have a few more guides are altogether become very essential for the cells if we cut there, but not everywhere. So we start to use a local smoothing model that take consideration of all the guide RNA in the neighborhood and then create a projection of the smoothened curve for each amino acid, indicating their potential CRISPR score of each amino acid. When we play with that, we start to realize maybe we can put these result onto the three-dimensional structure of this protein. For example, this DAR1L, the KMT lysine methyltransferase core domain was solved before. So we put our CRISPR result showing as the most depleted region as red hot, the less or non-depleted region as blue. And then when we start to look into this three-dimensional structure, we start to realize there are regions seems to be CRISPR hot pocket. And if we zoom in, we actually find within this hot pocket, there's a yellow molecule, which is the co-crystal enzyme substrate called SAMe, which utilized by this enzyme core as a methyl group donor to deliver the methylation onto the histone or target. And we also find that all the amino acids surrounding this same substrate interaction and their side chain are CRISPR hot. So this gives us an idea, maybe CRISPR domain scan maybe actually provide higher resolution than just define the domain boundary. We might be able to look into different portions of the domain and then how functional they are and help us to move forward the project. We also find another example in DAR1L, which is the AF9 cofactor binding domain as shown here. And this blue and red fragment is the DAR1L in this region, the most depleted region showing red, the non-depleted region showing blue at the corner. The gold color is the AF9 cofactor, and this is an NMR structure. So you can start to see the top 10 post of this fragment and showing the blue region is actually the tails, not directly bind with the AF9, and they are more floating. But the stable binding region to the AF9 is where the most important region of this fragment. So we think this method of a CRISPR tiling scan can also help us to zoom into the protein-protein interaction. Another example we try to scan in MLL leukemia is the LSD1 or KDM1A, which is an important demethylase that with a good clinical trial move forward in the patient. So we are interested in this scanning this LSD1 in MLL leukemia also. Therefore, we build a library of around 380 guide RNA tiling this protein with about 2.6 amino acid per guide RNA. And the result as shown similar way as the DAR1L, and we showed here for the LSD1 peptide annotation and their each individual guide RNA and the smooth and curve of the CRISPR result. When we look into this region, we realize the C-terminal half of the LSD1 is structured. N-terminal is kind of disordered. So we look into this structure, and then we plot the CRISPR result on top of them. Similarly, the red hot means the depleted region, the blue means the non-depleted region. When we look into the blue region here, actually we find this region is the soft linker between the N-terminal half and C-terminal half of this demethylase core domain. And it doesn't even have structure, and it's relatively tolerate to CRISPR mutation. However, if we look into the core domain of this enzyme core and start to zoom in here, there's a molecule co-crystal with this structure. It's actually the cofactor FAD. This is required for demethylase to propagate the catalytic activity. And we find that all the amino acids surrounding and stabilizing the FAD interaction are CRISPR hot, as showing at the arrows pointing here. Another example we find is the DNA binding domain SWAN, which is a smaller structure right here, just N-terminal to the demethylase domain. What we find that is this domain consists of 5 alpha helix, as showing as an example of SWAN domain from AD2 gene. You can see that they are the alpha 2 and alpha 5 helix are the two domains actually approach to the DNA and recognize the base pairs. The backbone alpha 3 does not touch the DNA. And when we look into our result, we actually find the alpha 2, alpha 5 helix are the CRISPR hot region, and alpha 3 is CRISPR cold. So this allowed us to start to think maybe we can prop the protein domain function using CRISPR tiling in a more dimensional, more variety of the domain function. So after that, we start to think about what's the limitation of the CRISPR domain scan. here we are seeing that usually when we do the depletion screen, we need to have cell number change. That means either the cell need to die or increase the proliferation. And this usually takes longer time to achieve the significant change of the cell numbers. And also it seems to limit to get the direct mechanistic information out of other than the cell survival phenotype. Therefore, there are more studies that we need to do. We are kind of interested to develop a way that can we link the genotype, which triggers by CRISPR mutation, and the phenotype could be the transcriptomic change. Therefore, we pay our attention into the single cell pipeline that provided by 10x Genomics. What we usually know is the 10x can have each gel beads have the cell barcode and then can capture the mRNA from each single cells, put on the barcode, and we know how they express in each single cells. And the newer version of the 10x genomic reagents also include several capture sequence. For example, the capture sequence 1. This allowed the users to start capture something in addition to mRNA. For example, you can buy antibody from the vendors that has their antibody conjugated with a barcode. You know this antibody based on the barcode, and also the capture sequence. Therefore, it can be incorporated into the single cell platform here and share the same single cell barcode. Recently, John Weissman's lab and then Britt Edison, they published a strategy to utilize these 10x genomics reagents for single cell CRISPR screen. What they propose is to include a capture sequence by the end or in the middle of the guide RNA. Therefore, these guide RNA can also be captured by the 10x reagent and then prepared as a library for sequencing. So we adapt this method and then move on to the high resolution way to characterize DAR1L's function using this capture sequence. So here, similar way, we designed around 600 guide RNA to saturate targeting DAR1L and clone into our improved version guide RNA with the capture sequence in the end. And then go through the single cell platform. In the end, we actually can sequence not just the guide RNA expressed in each cell and what are the transcriptomic signature from each cell. The result actually looks like this. Here, each dot indicates one cell in the pooled screen when we screen the DAR1L in the high density way. We can find around 7 different clusters of cells that shares slightly different transcriptomic signature. When we look into the marker gene expression, for example, the myeloid, the leukemia genes, the MEIS1, HOXA9, or the MYC oncogenes, they typically cluster to the upper right corner of this platform, the spectrum. And then if we look into the myeloid genes like CD11, GR1, or lactectoferrin, these genes tend to express in the cells clustered to the left corner of the spectrum. And then by doing the pseudo-time projection, we can actually predict the cell types are transition from leukemia to myeloid in this curve. What makes us excited is when we look into each single cell and the guide RNA expressed in each single cell, we can find that if we look into the guide targeting the enzyme core domain, which we know when knockout, the DAR1L will trigger cell differentiation, we find most of the single cell, including the enzyme core domain guides, are closer to the myeloid corner. On the other hand, if the guide RNA targeting the C-terminal or non-essential region of DAR1L, most of the cells having these guide RNA stays at the leukemia corner and similar to the control guides. So based on this, we start to do the domain analysis. Here, this heat map is actually a transcriptomic correlation heat map. It looks similar to the DNA structure, three-dimensional structure domain mapping, but here is actually we build the correlation of a protein DAR1L from N to C terminal of each amino acid and their transcriptomic signature correlation with each position on the same protein. Here you can see the enzyme core domain is right here. They have several internal correlation hotspots, so therefore we see a lot of connection here. We also find this enzyme core can actually work with a position we call the R domain is outside of the enzyme core and still work out very similar transcriptomic change when we hit it. And it actually reflects to this region when we hit this R1 region, the cell couldn't tolerate and will die eventually. Another benefit, I think, for the single cell platform is this screen in the single cell was actually performed just 3 days after deliver the CRISPR system. Compared to the traditional survival screen, by day 3, we barely start to see the phenotype, but the cells are relatively fine. We need a much longer then to start to see the cell killing effect. So this allowed us to have a chance to approach the earlier phenotype and then the driving changes at the early stage when we challenge this gene knockout, and allowing us to try to prop for the driver or causative mechanism. I think also this system allowed us to test some of the phenotype. In the end, it doesn't trigger the cell death, for example. It is a lineage commitment or the metabolism change. In the end, we also try to put the CRISPR, single cell CRISPR result onto 3D structure. So here we use the leukemia versus the myeloid signature as the y-axis, and then showing similar the smoothing curve. This allowed us to detect several regions are highly sensitive and when CRISPR will trigger leukemia differentiation. And we can plot these regions onto the 3D structure. Here is the 3D structure of DAR1L enzyme core domain together with the nucleosome. And what we can see is not just the enzyme core domain that binding the substrate, we also recognize a loop that stabilizes the enzyme and the target position interaction. We also can detect the region that stabilize the interaction with the histone H4 tail. A helix interact with the H2B, ubiquitinated H2, and then also a loop that stabilizes the interaction with the acetate patch on the nucleosome. So this makes us to think this single cell CRISPR screen provide a slightly higher resolution compared to the traditional survival screen. So here I would like to summarize that what we think the single cell tiling or assay tiling multi-omic screen that we provide the strategy to use the high resolution CRISPR tiling to prop a single gene. We utilize combined with the single cell platform that allowed us to capture both mRNA and the guide RNA for the CRISPR. We also can combine with the 3D data, structure annotation of the data. And in the end, we think we want to combine with the consortium genomic database that allowed us to identify more functional elements within in the patient cohort. And in the end, I also think that in the future, we can utilize the antibody to give one more layer of the multi-omics. So with that, I would like to thank all my heroes in the lab. Without their hard work, we don't have the platform right now. Our platform starts from microarray all the way to the sequencing, allowed us to identify different targets or zoom into different domains. And we are now in the process of trying to convert this pipeline to become a drug discovery pipeline. And I would like also thank all the funding support for this project. Thank you.
Thank you so much, David, for that wonderful talk. We have some time before the top of the hour. Please, if you have questions, please put them in the chat and address them to all panelists. I'll start with a question to David. So this is from Piyush Joshi. In your .1L screen, are you mutating each amino acid or pairs of amino acids? Or do you get functional protein for all editings except the ones that fall in conserved regions?
Okay, so I try to understand the question is what type of the mutation we got?
Yeah, so the first part was, are you mutating each amino acid or is it pairs of amino acids? Perhaps that's based on the number of, like the average nucleotides per edit that you showed.
Oh yeah. So actually it is actually a little bit more dirtier than that. So we use the CRISPR to cut different positions along the coding region. Each cut actually create a bounding hole. If I use that way to describe how diverse the mutation it can actually trigger by any of the CRISPR cut and repair. So, but what we find is it usually centered around where it cut has the highest mutation frequency, but it's quite diverse. So I think to answer this question, what we need to do is to do a deep coverage of the screen. Typically, we do 1,000x coverage. That means each guide RNA in average, we have at least 1,000 single cells in the library are carry the same guides. Therefore, give you a more sum up mutation profile, and then to know what's the general phenotype this can trigger. So we actually don't define the mutation in the clean way in this screen assay.
Okay, and so there was a second part to that question that I think is looking at the kind of the translational consequence, and that is, do you get functional protein for all editings except for the ones that fall in conserved regions?
If we got the functional protein,
maybe that's the question of whether you're creating missense or indel mutations for the editing.
Yeah. So I think it will all be there. So we do observe that about typically twofold depletion, 2 to 5-fold depletion will naturally happen. Doesn't matter where we cut for essential genes. What we are looking into is the region that cause much stronger depletion phenotype, for example, 100-fold, few 100-fold depletion. So those are kind of cannot be easily explained by those frame shift alleles, which exist in the pool, but we look for the regions that even we just slightly change the amino acid sequence could cause a very strong phenotype. So the point is this CRISPR tiling scan is if I say it's a domain discovery tool. After we find the region of interest, we will typically still go back to clone the whole full-length cDNA and then do the clean point mutagenesis of the region that we are interested, therefore define which amino acid is actually the driver for the phenotype.
Okay, thanks, David. We have more questions, but it's almost at the top of the hour. So here I'll stop this part of the session. I thank both of our speakers again. Drs. Chang and Wang, for your wonderful talks. And please, again, if you'd like to stay for continued discussion, please do so. For those of you that are now leaving, please join us next week for our next talk on May the 5th, where our own Jun Yang will be presenting Unraveling the Heterogeneity of Acute Lymphoblastic Leukemia through the lens of pharmacogenomics. So there'll be another wonderful talk, and please join us for that. So thank you again. So then with that, let's continue with Q&A. Liling, if I can ask you a couple of questions coming back to the liquid-liquid phase separation and the consequences of the mutation. So if I understand what you showed, the mutations are variable, but they seem fairly stereotyped. And if I saw it correctly, is it an introduction of a histidine, and that's what you mutated out that abolished the effect? It was changing the histidine. And so what do you think the role of that is? Is it binding metals, or is it how is it affecting that loop or beta strand?
Right. So you're right that there is so for the insertion, there is 3 amino acid insertion. It does have a histidine. We do not think it's specific to histidine. We think it's the insertion of the 3 amino acid as a whole that leads to the extension of the beta strand that is important.
Okay.
It can answer your question.
Mm-hmm. Okay. And I appreciated the word of caution you raised again about the level of expression. And so this, I know this is a challenge in the field, and we wrestle with it as well when we think about leukemia fusion oncoproteins and making sure that you've got that appropriate level of expression. So how are you approaching this issue in terms of, moving from the standard kind of workhorse that many use, the 293T or similar cell line, and then, making sure you've got an autochthonous model that's really addressing the question in patient samples?
Right. Yes. So we've been trying really hard to get patient samples. I have to say, if Sanju has anything, I'm happy to talk. But it's very hard to find patient samples that has this mutation, just because Worms tumor is very rare. What we have done is that we take the patient cell line, the Worms tumor cancer cell lines, and then compare the expression level of the ENL protein in those contexts versus our experimental system. And I wanted to say that what we're using in the 293 is actually similar level as what the natural protein level of ENL in those contexts as well. But on top of that, we also currently generate conditional knock-in mouse model for this mutation, so that this is an inducible system where then we can actually express the mutant under the endogenous promoter of the wild-type protein. And there, we actually see very striking phenotype in terms of perturbing gene regulation as well. So I think future work would be very interesting to move, as you said, to hopefully we find a patient sample or maybe in the genetic mouse model as well.
Okay. What are the downstream consequences of the condensate formation? Is there recruitment of new binding partners? Does it change target DNA binding site engagement?
That's a great question. That was actually a project that a graduate student who was actually a technician in Charlie Roberts' lab before. She's really interested in these questions. What we think, we hypothesize, this is to works that sort of more future works. We are interested in understanding, as you said, whether there's new partner are here. And this will probably involve, we're thinking about how can we actually identify those partners at endogenous contact, which is also very challenging, I guess, in the field. But on top of the new partner, I think that was really important to think about this potential pathogenic condensates that they might change the behavior even with the existing partner. So like maybe instead of doing what they normally do at highly dynamic, they might starting to actually behave abnormal, maybe leading to extended longer time of chromatin residence time, or maybe they have a higher frequency of interacting to the same sites, therefore might changing some of their transcription behavior, such as bursting and also the amplitude of a transcription events there. Another hypothesis we have, which I think is sort of inspired by people in the field who study this, is that we observe very site-specific condensate formation for this mutation, right? So what really drive those specific sites to be highly sensitive to this mechanism? And will this offer a way for cancer cells to actually, optimize its resources to highly activate certain subset of target gene instead of just globally amplifying everything? So, I think those are really the possibility, and it's still ongoing works. And I think that with the system, as well as other systems such as the one that you study with the oncofusion protein, we might actually start to get into by studying what happened in cancer, we can also feedback to what really happened in a normal as well. So I'm very excited about this aspect.
Yeah, as you pointed out, there's a lot to be discovered. It's an exciting time. David,
Yeah.
a question for you. Can you comment about where the field is headed in terms of modifying the nucleic acid sequence? So you've mentioned CRISPR-based editing, and obviously, I guess the original Cas9 enzymes can't modify every single base or perhaps every single amino acid. What are you thinking about different approaches, different Cas enzymes, different base editing, or are you taking this in different directions?
Oh, yes. Oh, this is a great question. I think, yeah, we are just start to touch actually the power of using CRISPR to test the domain function. So all of the example you described using base editor, using a different type of modified Cas has different pain requirement or higher fidelity, more targeting positions, will all give us different tool to look into this new field. I think base editor is for sure somebody wants to try that. Even though I think there are it has to be tested to know how efficient it's working. For example, what we learned from these so far using the standard Cas9 is we do want to have a very efficient editing efficiency. Therefore, we have much better signal to noise ratio to define the domain boundary, and also within the domain, where is the more important region. If the newer Cas9, even higher fidelity, but with a trade-off a slightly reduced editing efficiency, we'll have to see how the result, how easy to interpret the result. That's my guess. so far we've worked on this model and tried to screen for more proteins. I think my lab screened for more than 100 proteins already. We start to learn how to interpret this result. So, but we are very excited to move on to the new systems that hopefully provide different vision into the protein domains. Yeah, I think each of them will provide some different functions to help us in the future.
Okay. So another question for you, Liling. The number of punctures that you see in the T1 mutation are around 10 to 20. Do you see this also reflected in the number of ENL targets that are being positively regulated? Is there a correlation between the level of upregulation and puncture intensity or presence or absence?
That's a great question, and that was actually the first sort of surprising results that we had when Lola first saw this imaging. So short answer is that the one that I demonstrated to you, HOX, and also for several other genes that we have used FISH to validate, they are the very top positive regulate genes, and we noticed that they do actually co-localize with a hub. However, there are more ENL target genes that are positively regulated at the chromatin occupancy and also gene activation level than the hub that we are seeing. So there are several possibilities. One is that can one hub actually contain multiple target sites, right? I mentioned about those hubs of 500 nanometer size. This is actually incredibly large when you're thinking about when you compare to any reported transcriptional hubs out there. So we term this pathogenic elongating elongation condensate. Maybe they're doing something that is beyond what we typically think in a normal condition. And second is that perhaps here we're really only looking at very simple confocal imaging, right? So the 10 or 20 hubs I shown is the most notable hubs. But by higher resolution imaging, maybe we'll see more hubs, but smaller hubs. And this is indeed sort of observed by the chip sequencing that there are certain very small subset targeting has an incredibly high fold change in chromatin occupancy, whereas others has a more minor phenotype. So maybe this will be reflecting in terms of the resolution of the hub formation and sizes as well. A great question.
The same person asked that question said, so does that mean enhancer sharing?
Well, I would say genomic element sharing, not necessarily enhancer, because ENL typically don't bind to enhancer. It is more promoter focus region, which is very different than, for example, BRD4. But yeah, so this concept of potentially with this aberrant condensate information might help to bring things coming together. It's something that we're very exciting to look into.
Okay, thanks. David, if I can ask you a question. So, just thinking about the kind of fusion oncoprotein scanning question as well, that there are some limitations in that many of our fusions of interest don't kind of linearly give you nice in vitro transformation in a liquid culture that you can then kind of look for depletion. So how do you think about that? Have you done screens that perhaps look for a selective advantage that maybe expression of the fusion alone doesn't transform, but with the tiling approach you can then get outgrowth and you can read out mutations in that way? Or how are you thinking about perhaps extending the impact of this to those more difficult fusions?
Okay, yeah, this is a good question. So the short answer is I don't think we tried that yet. We did try like the MLL fusions in existing like leukemia cells and then be able to define functional domains. So those are clear drivers. If some of the driver just like give you survival advantage, or after longer term they can start to gain the advantage numbers, it will represent a more challenging samples for screen. The reason is we need enough time to allow the biology to evolve with bigger separation. But the longer of the screen, typically we start to acquire noise and the data variation start to also grow. So there's always a limitation how far we can go. But still, I think there's a possibility we can see some regions are more sensitive in the longer term of the culture. It's probably what we need to do.
Okay. Well, we're at the end of the questions that people have posted. So unless you've got questions for each other, we've got some time.
Yes.
Do you wanted to ask David a question? It's very striking when you do the single cell coupled with the CRISPR tiling that you know, even for example in the .1L itself, you were able to cluster region based on their transcription, single cell transcription, that, you know, perturbing one region clustered together, perturbing another region. So I wanted to asking the underlying observation in terms of what separates those two groups. Is it the degree of the same genes that are affected, like some region affecting more and some region affecting, but they're affecting the same genes, or they actually have very different gene targets by perturbing different parts of that one? Because if that's the case, it would be it's beyond our imagination initially, right? What's your comment on that?
I like this question. So because this is all the guys targeting the same gene, right? So I think I follow your question is they should somehow look similar because we are targeting, we cause the problem for the same genes, right? So that's actually what's the we observed also is they will share similar gene transcription change when we target several different regions in the same protein, but they don't always be the same. So for example, we actually find regions when they actually can work with the enzyme core that even it's outside the enzyme core, the transcription change looks very similar to when the guides target enzyme core. We find when the guide target the AF9 binding domain, it looks similar, but somehow more different. The correlation is weaker to the enzyme core. And what in the end we find is targeting AF9 doesn't destroy the K79 methylation on chromatin. Okay, so we still have enzyme activity. Targeting the R region that I described working with the enzyme core also cause loss of K79 methylation. So we believe that R region is actually a self-regulatory domain that we actually have a simulation the post interact with enzyme core. So it's almost like a SH2 domain in the kinase. It is a self-regulatory open closed region. And as a readout, it's an enzyme activity change. So we start to see different possibilities actually came out from transcriptomic signature that allowed us to learn sometimes one more layer of the mechanism. Yeah, but
Really cool.
I like this question. Yeah, and I actually have a question for leading also. Is for your I try to remember you do the IDR1 and IDR2, and they are actually opposing charges, right? In the normal condition. When you try to mutate and then cause the phenotype, how robust, how many mutations you need to introduce at each one to actually destroy those charge interaction?
So we have done 17 residues in the IDL1 region to blocking the charge. It's about 50% of other charge residues there. We haven't gone lower, which so that to your questions that we don't know how minimum that could be. Although I wanted to mention that I didn't have time to talk, but we also identified polar residues, for example, serine, that is very important in the IDR2. And in there, we're able to mutate it up to 2 amino acid or residues that can actually recapitulate the whole deletion of IDR2. So it's case by case. I think in terms of charge, it probably is a dosage dependent function. And in terms of other specific features, there might be a way. And I think this is a very good question also is because, you know, with IDR, it's sometimes, you know, if you're lucky, there could be very specific point mutation that can blocking the function of IDR. But in most of the time, I think that it's very hard compared with, you know, our, you know, long study structure lock and key interaction where you have 1 point mutation that does the job. IDR is sort of hard to perturb in a highly specific manner, and this is also one of the reasons we think that their ENL mutation, it's kind of a nice model to really for us to tease out the causality, because we were able to introduce very highly specific mutation that can affecting only the gain of function condensate, but not any other known function of this protein. But yeah.
Yes, thank you.
Great. Well, I think that brings us to the end. So thank you again. Thanks on behalf
Thank
of
you very
Charlie
much,
and myself
Charles.
and the Cancer Center, and it was good to see you both, and hopefully we'll catch up soon. Thank you everyone for joining.
Yes, thank you so much. Yeah.